Welcome to the EarthWorks Podcast, where our team will share the jargon of carbon with many of the turf friends we've met over the past 35 years.
Hi, everybody. This is Joel Simmons, and I got to say, I just recorded maybe my favorite podcast. Jack Higgins and I had a chance to interview Dr. James White from Rutgers University. And if you are a podcast listener, you've probably heard us talk about him. Dr. White is the person who came up with the model of rhizophagy. And we had the great pleasure and excitement to get Dr. White on our podcast to really go through what rhizophagy is in great detail. And it was fascinating. We were like a bunch of little kids all excited for Halloween or something because we got a chance to talk to our hero, Dr. White. And he went through the details of what this is all about. And it really fits perfectly into what we've been talking about for so long, biological soil management. So please, take a listen to the podcast with Dr. James White from Rutgers University and Jack Higgins and me. Thank you. Hi, everybody. This is Joel Simmons along with Jack Higgins, and we are incredibly excited to have Dr. James White with us from Rutgers University. We have talked a few times here on the podcast about rhizophagy, and it is probably one of the most exciting things that we have come across in
the last couple of years,
18 years that you've been here.
Yeah, sure.
And Dr. White has been really the breakthrough of this whole technology. And Dr. White, if you're okay with it, I'm going to read this real quick because I want to explain a little bit about what rhizophagy is. And I found this quote. I believe this probably is your words, but apologize if it's not. But rhizophagy is groundbreaking process where plants actively, and I love this word, consume and extract nutrients from soil bacteria and fungi. So we have known for a long time, I mean, we are, really trying to coin the phrase biological soil management. Our entire work and passion has been around feeding microbes through carbon-based fertilizers and things of that nature. But really, our passion has always been the biological, the biome and that whole underground world. And my understanding, and you can start off by correcting me if I'm wrong here, we only know about 5% of the soil biome. I mean, we've only really cataloged about 5%.
Well, I mean, that's a good question. I can't say the exact percentage. The point is, the point is that we cannot isolate most of the microbes that are out there in the world, in the soil and everywhere else. And we can do some non-culturing techniques to try to get some of them, and we get a lot of unknowns. We get things that we can get to the order, for example, or to the major grouping of microbes. But actually getting the species, knowing what species are there, knowing what they're doing, there's a huge, the popular word now is knowledge gap, right? A big knowledge gap.
Yes,
we
there
don't
is.
know. We really don't know.
Oh, is that a term that gets used in the professor world? Knowledge gap?
I think it is the knowledge gap. What are the knowledge gaps? There. So yeah. You write a review article. You want to address the knowledge gaps, for example. What's there, but we don't even know. You don't even know what you don't know. That's the problem, and that's kind of where rhizophagy came from. Really, is because people didn't know it was there, and the it's kind of like AI. AI can be a tremendous tool, but if something is totally undiscovered, AI is not going to discover it.
No,
that's right. It's only mining what's out there. So is that an accurate definition of rhizophagy?
It is those probably are from words of mine.
I'm guessing I stole it from you. Yeah, I'm credited.
But it's not. Yeah, but it's not. It's not the the technical definition would be a little more technical, right? I would add in that rhizophagy is a process where microbes alternate from a soil environment where they're acquiring nutrients and doing things out there in the soil to an endophytic environment where the plant can get those nutrients, inside the plant, where the plant can then get those nutrients and manage and use those microbes for whatever the plant needs, which is more than just nutrients. Actually, it's all kinds of growth effects and developmental effects and stress tolerance effects, and it's really a big deal, the process, and it's intracellular. It's not intercellular, like, some endophytes, we think, well, they go in between the cells and that they're really separate. But actually, these microbes in rhizophagy go inside the cells.
Oh,
inside
yeah.
plant cells. So that's really what, those are something that distinguishes that. And it's really a cycle, that it where the microbes are cycling between the soil environment and the internal and the plant environment inside the plant. So it's a
You're
alternation,
coming in and out. It truly
coming in
is.
and out, coming in and out, coming in and out. Yeah, coming in and out. So that really is the reason that, and all the benefits that plants get from microbes, is the reason that we want healthy soils. It's because the plants actually require those microbes. And we could put fertilizers on plants and grow them, and it's a great technology, but it has some side effects, and some pretty bad side effects to the environment. And once you start using excess chemistry in particular, then you have to use more chemistry. Once you start using nitrogen, for example, then you got to start, you enhance the fungal growth in the soil. It breaks down all your organics in the soil, and then you're feeding fungi that then could be pathogenic on the plant, and then you got to start using more fungicides, and it's just a never-ending thing. So,
Well, bringing up disease and disease resistance, something I've heard you say in talks before is that a healthy biological system, meaning like a whole big group of bacteria, everything like that, the full spectrum, they can actually essentially be a host for a pathogen. They can provide some of what the pathogen, say like the fusarium, is after. And so it doesn't have to attack the plant for that. Is that accurate?
yeah, that's accurate. The specific interactions are hard to put your finger on, but the fact is that when you have a good, diverse bacterial community, some pathogenic fungi like fusarium are not pathogenic, and those fungi will, instead of causing disease, they'll actually convert into endophytes in plants. And so you have, like in grasses,
Right.
fusarium is in it as an endophyte. Almost every grass, turf grasses, you could have, I spent years and years working on the epicloy endophytes in turf grasses, and tall fescue and ryegrass, and some other species of grasses, and there are fungal endophytes in there. Well, fusarium it's one of those fungi that go in, and that they go in when you, and non-pathogenic, when there's bacteria around to kind of regulate them.
Okay.
When do they become pathogenic? What creates that shift of
Well, I mean, you could probably figure it. If the plant gets under a whole lot of stress, and the drought stress, or any kind of metal stress or heat stress, then these endophytes, the fusarium endophytes, will revert. Stress in particular will do it. That so long as you have a nice mixed population of bacterium, fungi, bacterium, fusarium, or other fungi, and the system is healthy, the system is not under a lot of stress, then everything is fine. But once the system becomes very, very stressed, then that whole control mechanism breaks down. And we can do it in the laboratory very easily, just by growing seedlings, turfgrass seedlings, in a salt environment. And then what happens is the fusarium pops out.
You create the stress in the laboratory, and then that changes the path.
We can create the stress, yeah.
So
Yeah,
what you're suggesting to in our case, a turf manager is keeping the environment healthy, keeping a biological population strong. I mean, that's what's
that's
going to keep
correct.
that switch from switching.
That's correct. That's correct. I mean, that is really important. There's an analogy you could take, and that is, if you take the human gut, the human microbiome, like the gut microbiome,
I love this
we
analogy.
have, I know it
Yeah,
is, when we have
yeah.
a mixed population in there, balanced population, there's no disease. But if we, for example, take antibiotics and too many antibiotics, right, and knock out a lot of the bacteria, then we
Oh, the
suddenly
good
we start
guys.
having yeast infections. We have spore formers like Clostridium difficile, right? Bad gut microbe. Those microbes start taking over. Well, that's what happens in our soils, in our turf environment. Actually the root is comparable to a gut in that
Mhm.
respect. It's got
Yeah.
microbes in it and so forth, and interfaces with the soil environment when nutrients can be brought in. So uh, it is, yeah. I mean, the analogy with the human gut, which we're all aware of. Many of us are aware of how that disease happens there. Uh, it's similar, very similar to. And so you have systems where you over fertilize, for example, and then the whole system, you get a dysbiosis happening,
Mhm.
just like happens in the gut. You get fertilizer, or just treating the soil, treating plants and the soil like it's not a living system, like
Like it's
it's
a
not,
passive,
like
soluble
it's a
system.
passive, like it's rock wool or something, and the roots
Exactly.
go into it. It's just a substrate for roots to go into. That's not a correct analogy. That's not how soil really is.
And we still hear that story from, from a
Oh
lot
yeah.
of folks in the turf industry is that, soils are just a passive, holding site for plants.
Well, what I feel as though it prolongs the myth is that maybe for about 15% of the spectrum, that could be true, right? Like hydroponics, you can grow a hydroponic plant. It's not going to be a really healthy plant. It's not going to be a full,
That's right.
completely
That's
expressed
right.
plant, but
That's
but
right.
this but this but you
That's right.
can you can so like that
You
almost
can you can grow them like
you
that,
can grow.
and you
So that
can
almost
grow them.
prolongs this myth,
Yeah.
right?
Well, but you knew the side effect is you're using more chemistry on it.
Yeah, of
Using
course.
more chemistry, and
And you're
you know
getting a weaker plant.
you're getting a weaker plant. Exactly.
And even
You know,
mineralization isn't occurring because the microbes are bringing the minerals into the plant, right? I mean, isn't that part of the
right.
mineralization process, this whole rhizophagy cycle?
The rhizophagy microbes will loosen minerals in the soil, and they'll
Right.
acquire some of them, and some of them will just be directly solubilized, by the organic acids coming out of the root systems, and the whole system for bringing nutrients in. But if you don't have the microbe part, or if you just use fertilizer and try to bypass any of those other processes, yeah, you can grow plants like that, but then they're going to be, they're going to lack. You're going to get more deficiencies. They're going to lack some nutrients. They're going to be weaker. They're going to be uh, uh,
they
not
won't
stress
they won't
hardy.
stand up to all the rigors that we're
Yeah.
asking them to,
No.
especially in the turf grass world. I mean, we are asking
Yeah.
extreme rigors of these plants.
We're
Yeah,
cutting
mowing
them.
them. Yeah.
You're mowing them down to,
They're
in some
walking over them
cases,
every
on the
day.
green. On the
Right.
greens, we put them down to, oh my God, how
An eighth of an
oh,
inch.
Well,
the tiniest amount. And how can they tolerate that? They really have to be as hardy as they can be. And so, I mean, in my view, and this is very early on, and I know we love our turf, but there was a concept that's and I do too. I mean, I'm in the turf center. I don't know if I didn't
I,
no,
mention
we
that.
didn't.
it's on your shirt.
Actually, this might be a good opportunity to catch a little background about you
Yeah.
because we didn't hear how you got to where you are.
And your plant biology and plant pathology at Rutgers. That's
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 correct.
your position. How did you get to, what's your background? Give us a quick resume.
And then please remember your thought, because
Okay. Well, I don't know what that was, but
We'll
we'll
get
go.
we'll get back there.
We'll go now. Yeah. So I actually uh, got my PhD at the University of Texas, Austin, and I was working on endophytes, on fungal endophytes in plants. And I picked that project because from Auburn, I had known that that was important because they were just discovering in fescue grass. And so I was an undergraduate there and I worked on that. And I a little bit as a work study student, right, with one of the scientists. And then I went to University of Texas to work on the PhD. And so I said, well, I'm going to try to expand this, looking at wild grasses, other plants. And so that's what I did, 3.5 years. That's what I did. And in the process of doing that, I published a couple of things, and Reed Funk,
Mr.
who was
Rutgers.
Mr. Plant breeder, turf breeder at Rutgers, who originated the Kentucky bluegrass breeding program, really domesticated breeding, figured out how to breed Kentucky bluegrass.
Yeah, remarkable.
He actually took an interest in what I was doing. Sent one of the scientists from plant pathology department came to visit me in Texas, and some other people visited me. But from that early association, Reed Funk helped to create a position for me, a assistant professor position for me here at Rutgers in the plant pathology department. And so they hired me, and I came here and I started working on fungal endophytes of grasses, and that
To improve the genetics, I assume, correct?
to improve because the fungal endophytes were being discovered to have important positive role in
Are these
growing
mycorrhizae? Are fungal endophytes mycorrhizae? What is I mean,
no, in this case, no. There's another group of endophytes that are in the genus Epichloi. It's a fungal genus where they actually would go in the seeds and they'll vector through the seeds, and so you could put them in the seeds and then but they give the plant stress resistance, and they give the plant a lot more wear hardiness, and they extend the range, and insect resistance. That's the other thing, insect resistance. So Reed Funk started breeding them into turf grasses. And so they became, he actually originated the endophyte enhanced idea, putting the endophytes in there, enhancing the grasses. And that became something important. And so anyways, I came here and started working, and I worked on fungal endophytes, lot of different plants and a member of the Turf Center here. So I'm one of the ones that you don't see when you have the golf classic. You don't see me.
we don't see you,
me
but
at the golf classic. I don't play golf. I love the idea of golf, but
we do too, and we love your work. So this is all coming together.
Yeah, we're all on the same boat here. This is good. So you had started going down this road of these really low cut grasses and the stress that we're putting on turf, and and so what's the impact of all of that to this whole rhizophagy cycle? I mean, I'd love to hear this from your words.
So it is. So all of the stress we're putting on plants, putting on the turf grasses, makes them really susceptible to disease and just poor growth. Once they're under stress, when you cut them, they're going to be under more stress when you have excess heat and drought and so forth. But the endophytes, the rhizophagy is mostly bacteria, but also could be yeast. In some cases, algae get absorbed into the root at the root tip. And actually the plant will, we say farming, the plant will put exudates, sugars, and organic acids and other nutrients out there, and that will attract the microbes to the root.
We love that it's an intelligent decision by the plant to do this. We love that.
It's intelligence. What the heck, right? I mean, where's the brain? Well, Charles Darwin actually said the brain of a plant is in the root.
Wow.
So,
I didn't know that.
so,
That's good.
so yeah,
I'm going to write that down, actually.
that's a quote from Charles Darwin.
You never thought you'd get that on the
Never.
EarthWorks podcast,
Well,
did you?
yeah, from that. Exactly. But but this is happening in the roots, and the roots, there's a lot of interaction. There's I would say it really is a brain, but there's decisions that happen in those roots, and
Because they're
the
actually
plant
exudating stuff to attract and
attracts
colonize.
certain microbes that will
Wow.
benefit the plant. Yeah, certain now it's a very simple brain, right? It's a simple brain,
Right, like
but
mine.
well, like mine too.
Okay.
So, yeah,
comment.
but yeah, it's they attract the microbes, they secrete what they need. Like, for example, if they're low on nitrogen, they will withhold nitrogen compounds from that exudate. And
Oh.
and instead, microbes that can fix nitrogen and bring nitrogen from the air, whatever, they're attracted in and absorbed into the plant. And
Oh, that's fascinating.
So the plant can
I know.
actually regulate or ask for what it
the
needs.
the best evidence, it's simple, it's a simple system, but the best evidence that I've seen suggests that yes, they change exudates based on the deficiency symptoms, what's happening to the plant in
And my
terms of
understanding on that theory is that the plant will just continue to do that literally to the point of death, correct? I mean, if it's not finding the phosphorus or the nitrogen or whatever the mineral is that it's looking for, it'll just keep working that system until it's pretty much exhausted. Is that fair?
well, that's the cool thing about grasses, right? Grasses
Okay.
travel.
They
And
do,
so
they
they
move.
have something, they move, this
Mhm.
is why, back in the Cretaceous, way back, what was it, 70 million years ago, when the grasses developed, they spread all over,
Oh, yeah.
and
Oh,
they
and
just
you was on
moved
every continent.
every Antarctica, Antarctica, and it's full of endophytes, I got a
Wow.
graduate student working on Poania right now. It's a fantastic system.
Well, I wanted to get to Poa annua, and I wanted to get to
Yeah.
grasses in general, just
Yeah.
the way grasses behave,
Yeah.
because in one of your talks, I heard about how this process, and we're kind of still in the middle of talking about the actual rhizophagy taking it in now, taking
Yeah.
these
Yeah.
bacteria into the plant. But
Thank you for reminding me.
okay, okay. But so now we're at the point where the exudate has gone out, we've colonized what we want, we're taking it in. You use this example of, like, say we're trying to get nitrogen fixing bacteria into the plant. That now the bacteria come in, some are blasted, some with reactive oxygenating species,
Well, I'll tell you, you
oxidative.
get it.
So
Zach, you're putting us back on schedule,
okay, good.
Just go through that whole cycle so we
I
can get your
will.
definition
I
of
will.
top to
I
bottom.
will. I will. So they attract the microbes,
Mm-hmm.
and they will cultivate them around the root tip,
Mm-hmm.
and they will absorb some of those microbes into those cells at the tip of the root, at the growing. So every growing root tip is where this stuff is happening. And grasses have a lot of root tips.
Yeah, and you say that's like where the soft tip is. That's where
It's soft, because it's dividing, cells are dividing there, and they have soft walls, and so they can get them in. And they absorb them in, they take them in through those walls, through those very soft walls. And we don't know to what extent the microbe is secreting cellulases and other enzymes to get itself in, but we think that we know they get in because we can see them going in there. And then what happens is, once they're taken into the cells, taken actually into the root cells, the plant will hit them with superoxide, which is a form of reactive oxygen. It's a short-lived form of reactive oxygen, but it's very potent.
Okay.
And that superoxide will take away the cell walls on those bacteria.
And it doesn't kill them.
It does not kill them. Maybe some are degraded, but many of them survive. Once the cell walls are removed, then they become these protoplasts. And these protoplasts can last for a while inside these root cells, but they're also under control by the plant, because the plant is using that reactive oxygen to control them. The plant will actually multiply those protoplasts. So a few will come into the cell, it'll remove the walls and then make a lot of copies.
Well, they are farming. They're really
They
farming.
they really are farming these microbes. And so that definition is a very good definition for what is actually happening. The plant is replicating the microbes inside the root cells. And then this is the cool part, Jack. Okay.
Yeah.
I know you're ready to.
I'm ready.
You're ready for this. The cool part is they go into these epidermal cells that have the initials, it's a little bump on it.
Okay.
It's a root hair initial, and they accumulate in this little bump, that little root hair initial, and then they trigger the root hair to grow. Because if we don't have those bacteria there, we got no root hairs grow at all. And we could demonstrate that all day long, every experiment. We remove those microbes, you got no hair growth at all.
Wow.
So bacteria there, you get root hairs growing. And then as those hairs grow, the microbes those protoplasts are shuttled around and around inside those hairs, constantly going to the tip, accumulating at the tip, and then some of them will be caught and brought back and circulated around like that, go to the tip. And then at some point, a growth spurt will happen, and the microbes themselves will trigger the hair itself to grow, the ones at the tip. And when that growth happens, then some of those microbes at the tip, those protoplasts, are ejected out of the tip of the hair. So they go out of the hair tip back into the soil. And then when they go back out into the soil, right, as soon as they go out of the tip, they reform their cell walls. In fact, the plant will put, with those microbes, will put a little bit of exudate out, which gives them the energy.
Build the cell wall.
The cell walls, and if they have
Wow.
so they're soil competent. Right?
Of course.
So they're modified to be inside the plant, inside the root cells, and then they're re-empowered, redeveloped by the plant once it ejects them out to go back into the soil. And if they have flagella, little swimming thing, then those flagella reform. And so they can then go swim back out and acquire more nutrients and do whatever the microbes are going to do, and then get attracted back in to the grass. And turf is a champion of rhizophagy cycle.
That's what I'm thinking. Like it is
the champion of it.
Right,
Yeah.
because and why is that? Because it's just kind of it's low on the evolutionary scale. Like it requires
Botanically?
Yeah,
It is not. It actually is not low on the evolutionary scale. It is
Okay.
high on the evolutionary scale.
Oh,
It is
we've been
high.
wrong all these years. This is good.
It is designed. It is designed to colonize out into soil, and even if it encounters like sandy environment, to be able to go through that environment, right, with the tillers and rhizomes and whatever structures it's using to move across those environments, and it requires a lot of nutrients. And so rhizophagy is a way to get those nutrients, and it requires capacity for more root development. And
That's
when
right.
you have the microbes in the plant, you get a lot of roots. If you remove those microbes, you get very little roots. So really, growth is dependent on having those microbes. The plants are small and sickly without microbes. Yeah, you could pump them with nitrogen and get bigger plants, but you don't get the stress tolerance. You don't get the other benefits. There also is a effect that we're just seeing now, and that is that with my graduate student here working on POA annual, there may be another effect, and that is really a chlorophyll protective effect. when the
I took a note
yeah.
on bacteria keep chloroplasts from breaking down, endophytes
Yeah.
push evolution. Like you
Yeah.
said the bacteria
Yeah.
surround that
Yeah.
chloroplast. So
Well,
tell me more.
we've seen that, but we don't know if that's exactly what's causing the protection. We know that there's a great deal of oxidative stress protection, and that may extend. We don't know the details yet. That may extend all the way to the chloroplasts and the shoots. So while we see huge effects in roots, you could imagine, the chlorophyll and the productivity of the plant, the stress tolerance, the hardiness, of the plant, capability to grow, may require, in many cases, very oxidative resistant chloroplasts. And the endophytes and rhizophagy, it doesn't just stay in the roots, because what happens with rhizophagy, it's absorbing microbes from the soil. Those microbes go into phloem and into xylem, and they get transported in the plant. So it goes all over the plant. So really, the rhizophagy cycle, the root is the door to moving microbes into the rest of the plant. And
Just
so
like our stomach is, where it brings in our nutrients. Yeah.
So
like
for the benefit for several benefits, and one of them that your graduate student is isolating in on is this protecting of the chloroplast and maintaining color, it sounds like.
that's what we're looking at. And it's very preliminary now, but we know that we already know. It's not too surprising, because we already know that that with endophytes in general, you get more oxidative stress protection in the plant, and that
That makes sense.
it extends to chlorophyll and chloroplasts, and so for all parts, membranes. This is, if you Google that, you'll see that even in the Google.
So it's when the plant brings the microbe, it's the ROS that dissolves the microbe's body, and then that's when the harvesting of the mineral is taking place.
Yeah, what we think is, there's a couple of processes really. What we think is the cell walls are removed, and those cell walls have actually attachment sites on them for minerals and for other soil available nutrients that the microbe could use. But the plant gets them, and the plant takes the cell walls and deposits those cell walls inside the cells. So
interesting.
Interesting. It gets inside the cells, so we can actually see that.
So you see like remnants of cell wall and maybe mineral attached to it, you're saying?
Well, we can't see the mineral. No, we can't see the mineral.
But it's built from mineral.
It has minerals in it. They have nitrogen, for example, and phosphorus in cell walls, and
calcium, I imagine.
Calcium in there as well. There's different nutrients that are in microbial cell walls. So there's a whole bunch of stuff that go into cell walls. So there's really a lot there. But and what we do know is this: we could give different microbes will provide different profile of nutrients to the plant. So
makes sense.
They don't just take one microbe in; they take a bunch. And then they likely can select. And we talked about that for nitrogen already. Which microbes or favor certain microbes, right? But there's another process. Okay, so that's the direct process, and those are that oxidative component is probably more important for nutrients that are hard to get otherwise. That they take the microbes in and they oxidize them. And it seems like a lot of work for most nutrients, right? But some nutrients that are really hard to get, manganese, for example, or zinc or something like that, that's not freely available. They can then take the microbes, use the microbes to take it, and then take the microbe in, oxidize the nutrient out, and take it.
Right.
Okay. But when we do experiments using different microbes, what we find is that there's a lot of nutrients involved. Okay, but this is complicated because it's complexed by the fact that the microbes in the root cells are essential for plant growth. And if you don't give plants the microbes, then the roots are really underdeveloped. They don't have root hairs. They're not,
Like you
and
said.
we know the root hairs are for absorption. So to get the best development, the best nutrient relationships with the soil, those microbes need to be there. If you don't have it, if you pull, I don't know if of course you've pulled up grasses and can see instances where the roots just come out separate from the soil, and there's not a good interaction with soil.
That's right.
There's not. Okay, but when the microbes are there, that interaction, that association is tight, and you get soil adhering to those roots all around the roots, a nice rhizosphere.
Rhizosheath.
Rhizosheath idea, the rhizosheath concept.
Right.
Yeah. A rhizosheath, which is the soil adhering to those roots, and that rhizosheath is full of microbes. It's full. So not only what's happening at the tip where the plant is taking in microbes, but all along it, you have microbiology.
Now, those new root hairs that you mentioned that come from the rhizophagy, does that represent then a new root tip? Like the end of that root hair, does that count as a root tip, or is a root tip only at a kind of
Apical stem.
yeah, yeah, exactly, like a tap root,
It's like it's going to be if we pull a grass
It's
plant
called
up,
sure.
we're going to see lots of little roots hanging out there. Pull the soil off, you see roots hanging out there. All those tips that are sticking out that we see with the naked eye are all going to be root tips. The root hairs are going to be back behind those tips. And the little, if this is the root, it's going to be sticking back there like that. And we may not see that with the naked eye.
You can see it sometimes in like nursery plants, in like potted plants. Yeah, a little bit.
You could see them a little bit. You might need to squint down there to see. And it's the fuzz on the root. It's the fuzz on the root. That's what it is. It's the fuzz on that root. Those are root hairs. And the more bacteria you have there, the longer those hairs are. And that's important because we know that those hairs are absorbing other nutrients directly from the soil water, right? Where these roots are growing, and they're getting, so the more rhizophagy, the more microbe diversity we have, the longer those hairs are, the more dense the hairs on the roots are, and there's going to be more absorption. The more development of that rhizosheath, more nutrients coming out of that zone. And grasses are good. They can, they know how to travel.
They can do that
and get the fresh stuff, right?
We look at grasses as soil builders. I mean, grasses really are just building humus. They're building topsoil because they're like they're the super avenue for energy from the sun right down into that ground and kind of pumping it down in there.
Dr. White, let me ask you, how did you guys get to this point? I mean, what was the mechanism by which you were able to identify this? Is this all through microscope work? I mean, how are you actually mechanically observing all this activity in the soil?
It really is through microscope work. What we did, the first step was, actually we did a paper back in 2012 looking at the oxidative interactions on the grass roots. I think it was tall fescue and some other plants. Ryegrass, some other, maybe I don't remember what else we were looking at, but we started looking at how the interactions were happening with microbes and we developed some reactive oxygen staining techniques that we started using. And it's like when you put in this sense, that technique became like, I don't know if you remember when I was a kid, look at the comic books, and I was, 12.
Jack doesn't, but I do.
10 years old. You got these at the back of them, you have a little comic, a little advertisement for special
The
glasses
glasses,
that
yes.
you could put on and you could see through, with
Right.
see through in people's clothes or something.
That was so
Every
See
little
through
boy bought
walls,
them.
whatever it is, little boy thing,
yeah,
little,
right.
they sucked us kids in,
Right.
but
More
that
Cracker
crazy
Jacks.
stuff. They never had those glasses, of course.
No, I never got
It was
to see
$1
through
though.
anything.
You can get a pair for $1.
Right.
But this is like that.
Okay.
The reactive oxygen stains, let us look at those interactions,
Okay. So that's what started it.
right? That's what started it. And then we
Gotcha.
started seeing inside plants at this interaction. And we could see then that something is going on. There's some little blips in there, little spherical things in that. And then we started watching those and studying those and seeing where they could go out. And they were going out of the root hairs. We could see they're going out and they're remaking their walls. And they're becoming bacteria again. And then we started saying, well, my God, but where are they going in? What is going on? And so we studied that for a couple of years, trying to figure out that and playing with the systems and figuring out what was going on. And
Was there a major aha moment? I mean, was there a moment
There
when
was.
you guys were in
There
this and all of a sudden you realized?
There really was.
Oh, boy, let's hear it.
At a microscope. I was sitting with up in the lab at the microscope, looking at a system, looking at another grass, looking at was a Bermuda grass, and we were putting some bacteria in it. We were putting a bacterium that was a 4-celled bacterium, and we could see the 4 cells, at the root tips, where they were going in. We could see them going in, and then we could also see them once they went in. Then they became 1 cell, little spherical things once they were inside, and then we could see them at the root hairs coming out of the root hairs.
Wow.
In the root hair, they were little spheres, and then when they came out of the tip, they got 4-celled again. So they changed back, right?
Wow.
So we could see after we saw that, my technician at the time, Kate Kingsley, and I sitting there, we knew, oh my gosh, this is a one-way trip.
Wow.
It's a one-way trip.
It's a one-way
trip, and it's a cycle.
Yeah, right.
Because it goes around the soil, it goes in at the root tip, it goes out at the root hair, and then it could be attracted again back to the tip. So there, that became the
Aha moment.
what made it clear. And then we
Yeah.
started looking at systems, different grasses, different plants to see if we were seeing the same thing. And so it took us, we looked at this for a number of years before we finally published. So 2012 we started, right? Then we published the first paper on it in 2018. So that
Yeah.
was 6 years of working on it.
Wow.
And then after that publication,
That's the
then
one we
there was
read.
a lot of interest. Then I started talking about it, and we started studying, trying to figure out what are the, the details of the process. And we did, by the way, we did not invent this process. We didn't discover the process. We studied it and explained more about how it works. But the real discoverers were some Australians,
interesting.
Queensland, Australia. Yeah, in 2010, they published a paper in PLOS One, and it was like title was Turning the Table of Plants Consume Microbes as a Source of Nutrients. So
Wow.
these Australians, they really
Always
deserve
been a
credit.
big eco-ag culture in
Yeah.
Australia, right?
Yeah.
Jerry always went to Australia.
Yeah.
Yeah.
How long is the cycle? How long is it from when the microbes come in and then when they exit? I mean, what is it? Is it consistent?
It can be hours. It can be hours.
Okay.
It can be simply hours. It may be a little bit longer. It depends on how fast the roots are growing. In a fast growing root, it'll just be a couple of hours, and you'll see it. It'll go in at the root tip, and then you'll get hairs forming right away.
Wow,
and
that's
some of
pretty
them will
amazing.
come out. Some of them will come out at that root.
And then
So
some of them
so
stay in there, you said, and have a whole life cycle
they
and
they go,
repopulate.
and some of them stay in. Oh yeah, they don't all go at
Right.
once. the hairs would not grow at
Mhm.
all if all the microbes left, because the hairs only grow because there are microbes accumulating at the very
Oh,
tip.
because
It's
they're
the
forcing
accumulation.
themselves out. I see.
Accumulation at the tip, and then those are the ones that get ejected, but there's some on the sides so those at the tip are ejected. The ones around the sides that are circulating around the sides like that I'm talking about, they stay in there and they keep circulating. And what this circulation is really what breaks these microbes up, because
Yeah.
they're in the protoplast. And so they just keep breaking up into little smaller protoplasts, and those protoplasts get a little bigger. And so you just keep this internal
Cycle.
cultivation process. I mean, it really is the plants are farming
Yeah.
these microbes, and the root hair is actually a part of that farming process.
Talk to us about what happens in like a very highly sodic soil where bacteria would be less likely to proliferate. I mean, what happens in those types of really, toxic environments? And in terms of the cycle, I mean, is it not working well? I mean, is it affected? What happens?
Well, what happens in some of those environments is that plants will acquire microbes that are resistant to this, say, for example, sodium chloride, right? Salt
Yeah.
environment. So saline environment. And there are some microbes. In fact, what we can do is we can actually convert plants to more salt tolerant growth by giving them the right
Halophilic microbe.
halotolerant, whatever you want to call it, that can function in that kind of environment. And then you establish a rhizophagy process that can happen in that environment. Otherwise, it's a problem for plants. I mean, for example, if you consider like blueberries that grow in very acid soils, so acidity will reduce the growth of bacteria. And what happens there is that plants, when in that environment, will not form root hairs, because the microbes aren't there to go into the root hair initials and create longer hairs. But instead, what those plants will do is they will associate, like they will associate with fungi, that are able to grow in the acid environment.
Okay.
But at the same time, those are called ericoid mycorrhizae, by the way. They'll kind of replace the hairs completely. But if you take those plants that are adapted to using those ericoid mycorrhizae, you grow them in higher pH environments, then they'll revert to making root hairs again.
Amazing.
Wow.
It really is. It really, they are. I mean, it goes back to, and this is plant intelligence, we think of,
I was just going to say that. Yeah.
we think we are really the smartest creatures on the planet.
Exactly.
And we may be, but, grasses have their own, plants have their own kind of intelligence. And sometimes when we take over that control, of all the processes, we can do it.
We can do it better.
We can do it better. We can do it. But we create problems. We create some problems.
Of course.
That it might be better to figure out what the plants are doing and try to support that process.
That's exactly right. That's what we're trying to do.
Biological soil management in our mind.
That is it. Biological soil management. Consider what the plants are doing. I mean, we don't know all the details of it, and that's part of the issue, is that it's more complicated to manage the biology as opposed to merely putting chemistry.
Feeding the plant.
You can't think reductively, that's for sure.
No, you certainly can follow, you can follow a recipe if you're just going to use chemistry. You can follow a recipe and then deal with the side effects and so forth. And the side effects you can't deal with, then let them go, to the rest of the world.
Lose the grass, come back, get some new ones.
Whatever it is. But to ecologically cultivate turf is more difficult.
Yeah.
Now, I was going to say, which is one of the beautiful concepts uh, of golf, were these links courses, right? The
Let's
links
hear
course,
it.
the
Because I love
natural
them too. We love
courses,
them.
natural links courses. I have a picture somewhere that shows a links from St. Andrews, the links course, and
I worked there.
it'll work. Did you?
Yeah, 6 months I
Yeah.
was there. Yeah.
Wow, wow, wow. I was at Sports Turf Bingley many years ago. St. Ives. I was there many, many years. Reed Funk sent me there
Oh, get out.
many, many years ago. Yeah.
So anyway,
The
tell me
link- the
more.
link-
Okay, the links course.
That's a natural concept, and that is
Mhm.
those are on dunes and the worst environment. And of course, those grasses are able to, they're able to thrive there too. So it's not, it's not like it's impossible that we can manage plants, manage the biology. It's of course, when we stress them so much, that's hard for under any conditions, but the microbes being in the plant will make that stress more manageable.
Well,
for
and
the
the other thing I like about grasses is that they're perennial. So these microbes can actually train the plant to like evolve over time, and by, over time handle the environment, the difficult environment. And I think that's also happening, but from what you're saying, that they're
Well,
worth
that is happening. What's happening, I would say, I would put it as very, what you said is very insightful, but I would put it first, the plant trains the microbes.
Got
right?
it.
Plant gets the microbes it needs, it selects the ones that work with it, it grows. It really, so it selects a specific community that it needs, and it changed genetically. We haven't studied the details of it,
Mhm.
but you would expect that the microbes are going to be selected. At least the population is going to be selected, right? That's pretty clear.
Mhm.
The community will be selected of microbes is going to be selected for the ones that maximize the growth and survival of the grass, of the plants.
Perfect.
And the grass, it's got to be selecting that. But there is another effect which comes into the microbes training the plants, as you said. And it's very new, but what happens when these microbes are taken into the cells, they will actually go into, some cases, they'll go into the germ cells, like the pollen, egg cells, and so forth.
Wow.
But they'll accumulate around nuclei, and we can see that. We can see them going around the nuclei. And there is, we could also, in some of the work we're doing with the organic corn, with the microbes in the plant, they're going around the nuclei, but they're also causing more mutations.
They change the expression.
They're changing expression too. What they're doing is releasing methylation. They're demethylating, and methionine is an amino acid that's
They're
used
amino acids release, yeah.
on that's used on DNA to
Yeah.
turn off genes, right?
Oh,
And
interesting.
what
Okay.
they'll do, they'll demethylate genes and turn more genes on. So some of those genes that get turned on are the antioxidants like phenolics and
Wow.
other kinds of compounds that and other antioxidants. There's a lot going on, some of which we know. Probably most of it is, under this, like the
Mhm.
floating iceberg,
Yeah,
most of it.
right. I
Can't see it all.
We don't see it. We don't see that. What we know though is really interesting.
But you're saying like that the microbes are they're now deep in the nuclei, deep in the DNA, and they're turning on and turning off different.
This is what we think. We can see them around the nucleus, in the little nuclear envelope. What happens is when the cell divides around that meristem,
Mhm.
the cell, this is kind of just basic, biology. The cell, to divide a nucleus, it has to take the nuclear membrane apart to divide a cell, takes it apart, and then it'll take the DNA and separate it, right, to the chromosomes apart, and you have two cells. And then what happens is the bacteria, having entered those meristem cells, those dividing cells, it enters there. And then when that envelope comes back together, then it traps some of those
Wow.
bacteria in that envelope, right? And so
Yeah.
they end up there. And so is that the place where they affect the genes? That's what we think. That's where they're closest to the plant DNA. And this is really a process that's not very much known. And
Sounds like there's a lot that's not known, which is kind of
there's
the
almost
cool thing
everything.
about this.
Almost everything is not known.
Yeah, see, how is this? How is this whole concept accepted by your peers in the in the academic world? Are they adhering to this? I mean, are they understanding?
Some
Yeah.
are definitely
I've
some
seen
are.
that.
There are
It's
a
kinda
lot.
neat.
There are a lot. There are a lot. Some are.
Yeah,
Some don't know what to think.
yeah,
Right. Some don't know what to think. Some are. I mean, this is an endophyte process. So many are studying endophytes, and they're not thinking so much about the specifics of where those microbes are coming from, how they're getting in there, how they're getting processed. But
yeah,
it's everything takes time.
yeah,
It seems like there is more interest in the process from people in the regenerative
yeah, yeah.
agriculture,
Sure, biological focus.
biological farming areas,
Yeah.
that there's a lot more interest in, people who thought already that microbes were important think that rhizophagy process is really important, and endophytes are
Yeah,
really
yeah.
important.
Well, clearly,
But it's not on the radar
yeah.
screen of a lot of people.
You
And
had
why
said
should it be? They
right.
work on specific things. They're working on rhizobia or
Oh,
something
something. Yeah,
else.
Well, all growers want to get home to have supper, like they want the job to be easier. Like,
Yeah.
so they're interested in this.
yeah.
Whether you're maintenance greenkeeper or you're a farmer, you want the job to be more efficient and more successful.
And it sounds like that's what we're doing. You one of the talks I heard you talk about. You mentioned potassium being important in the rhizophagy cycle. Can you elaborate on that? I mean, what's the significance of potassium in this cycle process?
Well, potassium is one of the nutrients, right? It may
Mhm.
not be the most important in all plants, but we did an experiment in which we used carbon dioxide to suppress in tomatoes to suppress oxidative extraction from nutrients. And when we did that, potassium fell. Roots got tiny potassium levels fell.
Mm.
And so what and when we measure potassium, I'm talking about potassium per weight of the tissue, right? So
Volume.
it's comparing apples to apples, and not apples
Got
to
it.
oranges, it's so and that but it's really only one of the nutrients. It is important, but all of them, I think, many nutrients are important. The
Sure.
most important is, I think, and this was our idea from the very beginning, are going to be some of the micronutrients that plants cannot get easily from soils. That's going to be the most important. But I tell you what, rhizophagy it is not exclusively, and I've said this already, a nutrient process. It's
That's right.
developmental process. And you get bigger roots, you get bigger shoots, you get better growth, more branching, more root architecture, hairs for better absorption.
Plant can get water more
Plant
effectively.
can get water. Plant photosynthesis is going to be better.
Yep.
It's going to be more efficient. It's going to be more stress hardy, more disease tolerant. You're going to need less fertilizers,
Yeah.
less chemical
Good.
fertilizers. There's a lot of effects that, and that's all that added together. To me, it means it gives all the reason that we should try to grow plants, turf, using microorganisms,
Right.
favor the microbial growth in the plants, to build the organic material in the soils, which are essentially the home and the food base for the soil microbes. It gives all of that together. It's not just one little thing that we maybe, that's variable depending on the situation of the plant and the microbes that have to be involved, but the whole picture. And what it really does, and this is something that growers understand very well, it changes the paradigm. The paradigm that we had before, which is it's all chemistry. It's all chemistry. And it changes the paradigm to say, no, it's not all chemistry. This biology is critical. This biology. So maybe we should start thinking about that biology and start trying to grow our turf and grow our other crops using biology. That doesn't mean we have to abandon chemistry entirely, but we start to focus more on this biological environment in the soil that our plants are living in.
Let me ask you this too. Has genetics affected, all the work on genetics, certainly in the turf industry, and you're familiar with this, having worked with Reed Funk. The genetics that's going on in turf, is that affecting this cycle process? Are they impacting it in any way? Are there newer plants better? Are they worse? Are they the same?
Well, turf is a unique kind of crop in that it's still pretty close to Mother Nature, right? Original. Turf really is very close.
COG, love it.
We have not had a whole lot of genetic manipulation going on,
Nice.
at the especially at Rutgers, we occasionally we do some experiments here or there to turn this gene on or off, but by and large, the material that we grow is that our plant breeders are producing. Stacy Bonos is now our primary plant breeder. They're not genetically manipulated. Other than just what classic human selection process is.
Yeah.
And that's really by design. That the idea is that we want plants that are going to be acceptable as much as possible by a broad base of the population. And so it's oftentimes it's better to just select them,
find the ones that are doing the right things.
Yeah. And so we have not, and turf in general has not been modified as much as, say, a crop like corn, right? Corn.
How about the genetics in corn, though? Is that affecting this cycle?
Well, they say, there are articles about corn having lost its capacity to its NOD gene, for example, the NOD gene that's in there that may connect with mycorrhizae, so that some mycorrhizae cannot now connect to the corn roots.
Oh, wow.
That ain't good.
So
that's not good.
But that's a different, a different system. And, we also have corn that's not been modified like that. We have organic corn that there's problems with contamination, but like I work with a corn breeder, Walter Goldstein, Wisconsin.
Yep,
and,
I know of him.
You know of him, yeah.
Oh, yeah.
And he, his corn is microbial rich.
Yeah.
And where we see, we have microbes in the corn, that's where we see increased mutation, which means transposon, these jumping genes, moving genes around, and that's where you get more genetic variation. So that could be impacted in some of this other corn. But the other varieties, right, of corn that have been highly selected. But the other thing that we're seeing also is that it's not necessarily a death sentence to a variety that once it's selected for, maximum growth using fertilizers and chemicals, that's not necessarily a death sentence, because what we're seeing in Walter Goldstein's breeding program is that those plants can be remediated using microbiology. After 3 or 4 years, you grow them with microbes,
Clean
put
up the
some
system.
ferment on them and
Yeah.
clean them up, and they start to respond to the
Fascinating.
biology.
Well,
So they can be saved.
that had me thinking about breeders in general. I would imagine breeders want to breed then in a very microbially rich environment. Wouldn't you want to do this breeding work in soils that have a wide array of microbes to make this happen?
Yeah, it really is. That's a concept that, occasionally over the past 8 years or so, people talk about, and of course, Walter Goldstein has been doing it. He's
Right.
been doing it. But I've seen reference to it now, and I don't know where else I saw it, but other people are catching on. The use of biology in breeding programs to try to incorporate that into the varieties, so that you have, and they'll go onto the seeds and so forth, and people get seeds, they'll have microbes on there that are,
This
and
is like
you'll
the
have
end of
plants
fight.
that are
Yeah, the end of
plants that are trained
Mhm.
to deal with microbiology, so that's
That's kind of what I'm thinking.
yeah, oh yeah, that's it's beginning to happen. That
Yeah.
is beginning to happen.
Yeah, we've been doing this for 40 years, and I can certainly say that, the interest among growers has grown exponentially in the last 5, 10 years on this whole biological soil management concept. Dr. White, I hate to say this because I could go on for another hour or two, but we've run an hour of your time. But can you give us a little summarize this whole rhizophagy thing, and if just so that we have.
Or I guess like a summary, and also like make it what's the practical thing that these daily guys that also have to produce playing conditions and do the job, like what's the way forward to kind of utilize some of this strategy?
Well, that's a good question. It's actually hard to answer that. I would say the important thing to remember is, don't think of your soil as just an
Substrate.
inert substrate, an inert something that would a matrix that your plant roots will hang on and that your plants will grow on, but instead as a biological entity. Do treatments that will preserve the organic material in soils, build your organic material, um, uh, limit applications of substances that might damage that microbial community. It's and in terms of, okay, so those are a couple of things. I always say, build your soil, because that's the important. Build your microbial community. Think of that. Use of biostimulant microbes is beneficial. And those could be defined, bacillus, for example, and putting bacillus on seeds when you put it in, or putting your biostimulants on seeds. What that does is that will actually kickstart the rhizophagy cycle, and the microbes then start coming in like that. So you're using microbes in cultivation.
Right.
I'm not trying to sell any particular products, but some products are undefined. Those are good products too. When you put a lot of microbes, what I mean by that is it's a mix of microbes that you don't know what everything is there.
That's right.
All of the ferments that people are using, these are undefined. And that gives a lot of those work really well.
Yeah, fermentation has been an interesting process, certainly for us.
It just encourages an enormous microbial
population.
Yeah, population.
I mean, our take has always been just basically feed the microbiology population by giving them food
that's what I
sources.
say.
Yeah.
Grow your crops by cultivating the microbes, the
Yeah.
microbes.
Farm
in
the farm
plants
that's
and
being farmed by the plants.
take care of those microbes, and your plants will do better.
Perfect.
That's the key. Yeah, and the other thing is, what is the rhizophagy cycle? What is rhizophagy? The simplest way to think about it is, and I'll just say it by the word definition: rhizo for root, eating for phagy, rhizophagy, root eating. It's basically the process where grass roots, plant roots in general, eat soil microbes. They
Eat
don't
'em,
degrade them
baby.
completely, but they're
We're going to
eating
have to
them.
send you a Jack Higgins podcast because he's been
They're eating
using
them.
root eat for a long time
Root
since
eat.
hearing of your work.
I think about Little Shop of Horrors. Yeah,
Yeah,
right.
yeah. Well, we need those kind of analogies. It's rhizophagy. It's a funky word.
Mhm.
But it simply means root eating, and
Root eating, yeah.
that defines the kind of the process. And it's cyclic. Yes, it's cyclic, but some of them are degraded completely. So yeah,
Wow.
I
Dr.
think
White,
yeah.
I love that. That's just wonderful. I can't thank you enough. I can't tell you how excited we were to be able to talk to you. Thank you for responding, and thank you for all the work you're doing. And I hope that we can get you back down the road here and hear some of the newer things. I love the work you're doing with POA. I think that's brilliant.
I think that would be very cool.
Yeah, that would be fascinating. And you've been getting around. I mean, you've been getting around the talking circuit. We've certainly seen you here. We've seen you at Acres
And heard you on a number of podcasts.
Yeah.
We appreciate your communication. We appreciate you getting out there and talking with us and talking with everyone, because people are getting excited about this.
Yeah, it really is. It's a change in the industry, and I think there's definitely a shift, and you're definitely pushing this, and I'm grateful for you. Thank you.
Well, thank you, Joel and Jack, for inviting me here and saying all that good stuff. I really appreciate it. I really appreciate it. Thank you.
Right.
We mean it earnestly. I can promise you that.
And we hope you have a prosperous summertime. You mentioned you've got some work travel ahead, and back to education then, I'm sure, in the fall. And so we appreciate it.
Are you
Well, thank-
teaching this in classes at Rutgers?
I do teach it, but it's not a whole class in it.
Okay, no, I wouldn't think so, but it would be incorporated into a
Oh yeah,
plant biology class.
yeah. I teach
Beautiful.
it. Yeah, I do classes in fungi, courses in fungi. So I talk about it in
What
my college.
majors take your classes? Like what kids take?
Everybody.
Everybody.
Micro majors and plant pathologists and plant biologists, and and I have one course, poisonous and edible mushrooms, which
Oh,
has
great.
liberal arts people in it too.
I
Of
bet.
course.
I bet.
For a lot of reasons.
Sure, yeah, multiple reasons.
I taught high school. That's all they wanted to learn how to grow.
That's
Yeah.
it.
Are
Yeah.
you getting into the turf program at all in Rutgers, the 2-year program? Have you gotten to talk to those folks at all?
I think many years ago
Yeah, you
I
should.
gave a
We're
couple
going to
lectures.
encourage you to do
Yes,
that.
we want you talking to those folks, please.
Brainwash those
They'll
guys.
think I'm nuts.
Yeah.
Okay.
Again, thank you so much. We're
It's all right.
we've run more of your time than deserved, but I'm grateful and thank you so much for all the work you're doing. Appreciate it.
You're welcome.
And
Okay,
folks,
bye.
that is the EarthWorks podcast, one of our favorites so far, but stay tuned and we'll let you know when other folks come up. But hopefully we can get you back, Dr. White. It would be a real pleasure.
Thank you.
Thank you.